Let's talk a little bit about the modes of reproduction of bacteria. One of the things bacteria can do is this first one called transformation. And what we see in transformation is this is some DNA that's just kind of floating around in their environment, and they pick it up. They pick it up and they incorporate it into their own nucleic acid. And whatever genes are located on them, they now have those genes. One of the reasons why bacteria evolve resistance so easily is they pick up this stray DNA, and sometimes that stray DNA has ampicillin resistance and other kinds of resistant genes out there. Another way that we can get new DNA into a bacterial cell is through transduction. This little hexagon structure here is supposed to represent a virus. This virus has nucleic acid in it. It's going to inject that into the bacterial cell, and that bacterial cell is going to incorporate it here into its own DNA. And then finally we have conjugation. In conjugation, one bacterial cell will give some of its DNA to another bacterial cell. Now, some prokaryotes are harmful. You've heard of MRSA, methicillin-resistant Staphylococcus aureus. So these are bacteria that have become resistant to methicillin antibiotics. We also see that E. coli can contaminate food sources. Salmonella can also contaminate food. And botulism can cause disease. Now, botulism is kind of an interesting one. It can, under certain circumstances, be used beneficially. We can use the botulism toxin to create medications. For instance, migraines are often treated using Botox injections in areas where there's a lot of overactive nerves, and it causes those nerves to quit sending all of those pain signals, and it helps reduce the frequency and occurrence of migraine. So we also have other beneficial prokaryotes. Some are used, of course, to make various food items, cheese, yogurt. Many species of bacteria can also break up and degrade oil. In some big oil spills, like the Exxon Valdez spill, we can release these kinds of bacteria to break that down. We refer to it as bioremediation. We are using a biological organism to remediate. We also have a lot of beneficial microbiota in and on our bodies. We have a lot of beneficial microbes in our gut that help us digest food and works with our immune system. We have a lot on the surface of our skin. So there's a lot of good bacteria. And part of the problem with overuse of antibiotics is not only are we allowing things like MRSA to evolve because we are essentially creating a very strong selective pressure for those that can get around our antibiotics, but we are also, in many cases, killing off our good bacteria. That's one of the reasons why sometimes when you take an antibiotic, it's good to, at the same time and afterwards, take a probiotic to reintroduce good bacteria into your body. We have the prokaryotes, we're going to move into eukaryotes. Where did they come from? Now remember, one of the things that we find in eukaryotes is we have that membrane around the nucleus. We also have these other organelles that have membranes around them. And two of the really important ones are chloroplasts and mitochondria, because these are the energy harvesting organelles inside of cells. Chloroplasts perform photosynthesis, mitochondria perform aerobic cellular respiration. How did they arise? And so this is known as the endosymbiotic theory. We have essentially the precursor to a eukaryote. We have a prokaryotic cell. One of the first things that's going to happen in this prokaryotic cell is this membrane is going to start folding in on itself to create some of this endomembrane system that we see here. And then we're going to have an endosymbiotic event. What that means is this cell is now going to engulf another cell. First, it's going to engulf an aerobic bacterial cell. So this is another prokaryote that is performing aerobic metabolism. Essentially, that cell now becomes the mitochondria that we see inside of these eukaryotic cells. This cell then also can do a second endosymbiosis and ingest a photosynthetic bacteria. This photosynthetic bacteria is essentially going to give rise to chloroplasts. Here we would have the origin of our organisms that perform photosynthesis. Here we have our origin of organisms that do not perform photosynthesis. But both of these are now eukaryotic cells. Remember, we said when we get to eukaryotic cells, we're going to have sort of two major kinds of groups. We're going to have those that photosynthesize and those that are heterotrophs. They don't photosynthesize. Let's go over to protists. So protists are eukaryotic. They have these membrane-bound organelles. What are some of the other characteristics of protists? Well, I put a big question mark here because protists are tricky. Protists are basically the junk drawer of taxonomy. It's probably not a true single group. So when we make a group in taxonomy, we infer that that entire group has a common ancestor. And that probably is not true for protists. They are very diverse. Their structure is diverse. You can have anything from single-celled organisms. You can have these very large multicellular kelp beds. They're very different in the way they reproduce, different in the way that they obtain energy. So some are photosynthetic, some are heterotrophs. So they are just all over the place. Now, one of the things that protists do, if they are heterotrophic, they have to ingest food. This is showing how they ingest food. This is called phagocytosis. They're taking their cell membrane and pushing it out into like these little arms, pushing the cytoplasm out into it to wrap around this food particle. Or if this is one of your white blood cells, this may be debris, damaged tissue. It may be a pathogen. It's going to wrap around that, and we're going to create what we call a food vacuole. And then inside this cell is another sac that contains a bunch of digestive enzymes. And that is going to merge with the food vacuole, and that's going to allow this cell to break that down. It's going to use what it can, and then whatever it can't use, it'll essentially do the reverse of this phagocytosis. It'll come over here and it'll eject what it cannot use. Now this shows us the supergroups for the protists. Again, keep in mind that different books will show you different things because it really has not been resolved. We do not know exactly how to resolve the protist group. Some protists are pathogenic. Up here we see the organism that causes malaria. This Plasmodium, it's italicized, so that means it's a genus name. And when you see this SPP, it means that there's a variety of different species that can cause malaria. Rather than list them or identify them as individual species, we'll just say it's several different Plasmodium species, and that's how we'll do it. Trypanosoma, several different species can cause African sleeping sickness and Chagas disease. We also have some protists that are pathogenic to plants. We see our downy and powdery mildews on plants are caused by protists, and we also see the potato blight is caused by protists. Now, some protists, of course, are also beneficial. They form significant components of our food webs. A lot of plankton are protists, and they form the basis of the food web for aquatic environments. Protists account for about 25% of the world's photosynthesis. We also see that protists can be symbiotic organisms to others. Here we see an example of coral. Inside the coral, you have a photosynthetic protist that is living inside the coral. It's providing energy for the coral, and the coral is providing a place for that protist to live. You also see symbiotic relationships between termites and protists. Termites do not have the enzyme for breaking down cellulose, which is what wood is. They have these symbiotic organisms that live inside their guts that do that for them. And then, of course, a lot of protists are living in the soil, and they act as decomposers, returning dead and decaying organisms, releasing those nutrients back into the soil.